Imagine being born with a disease that can bring unbearable pain, without warning, over and over again, for your entire life. Now imagine scientists discovering a way to switch that disease off, not with years of medication, but by rewriting your body's own genetic instructions. It sounds like science fiction, but it's happening today. This extraordinary breakthrough earned Professor Stuart Orkin and Professor Swee Thein the prestigious Breakthrough Prize in Life Sciences, often called the Oscars of Science. Their decades of research have led to gene therapies that are giving people with sickle cell disease a completely new lease on life. But here's the heartbreaking part. The people who need these treatments the most are often the least able to access them. So, how did scientists achieve this remarkable breakthrough? And what happens next? For decades, doctors could only manage sickle cell disease. They couldn't cure it. People living with the condition often relied on pain medication, frequent blood transfusions, and sometimes risky bone marrow transplants. These treatments help people survive, but they never solved the root cause. Then, scientists noticed something remarkable. A small number of people naturally continued producing fetal haemoglobin, the special type of hemoglobin every baby uses before birth. Surprisingly, these people usually experienced much milder symptoms. That simple observation became one of the biggest clues in modern medicine. Scientists began asking a fascinating question. What if we could persuade every patient to make fetal hemoglobin again? Finding the answer became a scientific journey spanning more than 20 years. To understand this discovery, imagine your body is like a factory. When you're a baby, that factory produces one type of hemoglobin that carries oxygen extremely well. But shortly after birth, the factory receives new instructions and switches to producing adult hemoglobin instead. For people with sickle cell disease, that's when the trouble begins. Professor Swee Thein and Professor Stuart Orkin helped uncover the hidden genetic switch responsible for that change. The switch is called BCL11A. Now here's the surprising part. Instead of trying to repair damaged blood cells one by one, scientists realized they could simply turn off this genetic switch. The body then starts making healthy fetal hemoglobin again, protecting red blood cells from becoming the rigid, sickle-shaped cells that cause pain and organ damage. Sometimes, solving an enormous problem begins with finding one tiny switch. This breakthrough didn't happen overnight. Professor Swee Thein spent years studying families affected by beta thalassemia. One extraordinary family living in Malawi became especially important. She carefully traced 210 relatives across 7 generations, searching for inherited clues that explained why some family members naturally produced higher levels of fetal hemoglobin. Meanwhile, Professor Stuart Orkin tested the idea in specially engineered mice with sickle cell disease. When his team switched off the BCL11A gene, the disease was dramatically corrected. Then another scientific revolution arrived: CRISPR gene editing. Scientists could now precisely edit a patient's own blood-forming stem cells before returning them to the body, transforming decades of laboratory research into a real medical treatment. Every great discovery was built upon the one before it. The results have exceeded many scientists' expectations. The first approved treatments: Casgevy, developed by Vertex Pharmaceuticals and CRISPR Therapeutics, and Lyfgenia, originally developed by Bluebird Bio, have transformed lives. Clinical studies found that more than 90% of treated patients became functionally free from the devastating effects of their disease. Many people with sickle cell disease no longer experience the agonizing pain crises that had controlled their lives for years. Patients with beta thalassemia often no longer needed lifelong blood transfusions. Imagine finally being able to plan your future without constantly worrying about your next medical emergency. For many families, that future is becoming reality. But every great scientific breakthrough comes with difficult challenges. Today's treatments are extremely complex. Doctors must collect a patient's own stem cells, edit them in specialized laboratories, give intensive chemotherapy, and then return the corrected cells to the body. The total cost is estimated at between $2 and $3 million for just one patient. That's especially heartbreaking because sickle cell disease affects millions of people, particularly across sub-Saharan Africa and India, where these treatments remain largely out of reach. But scientists aren't stopping there. Professor Stuart Orkin hopes future patients could simply take a pill that switches fetal hemoglobin back on. Meanwhile, Professor Swee Thein is developing medicines that could prevent the painful crises themselves. The next revolution isn't simply about creating better treatments. It's about making those treatments available to everyone.
This remarkable story reminds us that the greatest scientific breakthroughs often begin with a simple question, followed by years of patience, persistence, and collaboration. By discovering how the BCL11A gene controls hemoglobin production, Professor Stuart Orkin and Swee Thein have helped change the future of sickle cell disease forever. Now, the world's next challenge is ensuring that this life-changing science reaches every patient who needs it, regardless of where they live or how much they can afford. What do you think? Should life-saving gene therapies become affordable and accessible to everyone around the world? Share your thoughts in the comments below. If you enjoyed this story, please like, share, and subscribe to Innovative Scientific Discovery. Thank you for watching, and I'll see you in our next scientific discovery.